All right, welcome everyone. Good morning. Just a heads up as you're signing in. I have done an audio check, thanks to those of you who helped me with that, but I'm a little concerned my headphones might be starting to die. So if you are not able to hear me at any point and yet my lips are moving, just say something in the chat right away, or even unmute and let me know. And I'll go back to the regular audio with no headphones. I just find on the recording that the headphones are much better. So you'll find there's less background noise. And also when my household is getting crazy and my toddler is having a complete meltdown, it's harder to hear that when I'm on my headphones for you guys, not for me. I can hear it just fine, which is what it is. Okay, let's go. So this is the second day of class. Welcome. Welcome if you're normally here for mornings because that's your section. Welcome if you're from the night class and this was better for you this week. Welcome if you're from Saturday and you're attending this Zoom. I just want to reiterate what my expectations are in general. For each topic, you want to prepare ahead. Now, today is just day 3 of the quarter, and it may not have been possible for you to do that. And that is just fine, but you're going to have oodles of time before next week when we meet again. So just as an example of what I would normally expect, today's topic is Introduction to Microbiology. How would you know that? You would look at the schedule to see what that topic is. And then before coming to this Zoom, you would have gone to that topic page, watched the prepared video lectures, read the textbook chapter, which I've said twice apparently. Oh yes, just as an example, like you would watch chapter 1 video lectures and read chapter 1 in your textbook. So what do we do in the Zoom? This part here of watching the pre-prepared video lectures is actually required. That's where all the information is. What we do in Zoom is we review some of the key concepts. We apply our knowledge, we expand on things. I give you a chance to study, review, apply, but it is not a substitute for watching the prepared video lectures. And at the same time, in my experience, the best academic outcomes are if you do this prep and then attend the Zoom live, you will get the best outcome in the class, usually, for most people. A reminder that the schedule does show you what topics are coming up so you can prepare ahead of time. And so that's my expectation is that, for example, next week, you're going to take some time, let's say over the weekend, or maybe earlier, depending on your work schedule, to do this preparation. Okay, so let's talk about chapter 1, kind of big picture. We're going to talk about what microbes actually are. We're going to define them. We're going to talk about the 8 types of microbes and then review some of the distinctive features. How can we tell the difference between them? Another question that's addressed in this chapter is, why do microbes matter? And so there's a lot of different answers to that. I asked you this question last class, and I'm going to re-ask it again just to see if in the intervening 2 days you have new answers. But it's not really important if you have brand new answers today or not. It's more important for you to think about this all quarter and develop a more sophisticated answer, a broader answer, a deeper answer by the end. That's the goal. We're going to talk about how we know about microbes, a little history, just a smidge. I'm not going to cover everything. You're going to get a taste of what kinds of information you need to have prepared for the test, but really the prepared video lectures are more complete. And we'll end with some of the current challenges in the field. So first, the definition. Microbes are defined as organisms and/or infectious agents that are too small to be seen. So organisms implies life, cellular life. So all cellular life that's too small to be seen is considered a microbe. So single-celled organisms, a few multi-celled that have not very many cells are microscopic. And infectious agents that are not organisms are these non-living guys that don't have a cellular composition. They're not made of cells. They're smaller than cells, and yet they are able to infect, cause disease, and they are able to replicate. That's a really important component. So here's just a few examples. Here's a bacterium, which is a prokaryote. There's 3 different species, as you can see in this pseudocolored electron micrograph. Here is a protozoan parasite, Plasmodium, a causative agent of malaria, living on the mosquito intestine before the mosquito goes to bite you and inject it into your body. Here is a microscopic fungus. So this is one of the eukaryotes that is somewhat multi-celled, but nonetheless is microscopic. So a mold basically on a medical implant. And of course, our good friend from the last few years that we can't get enough of, coronavirus, in an electron micrograph as well. Now, one of the things that's challenging about this field is the size of everything. So it's hard because unlike anatomy and physiology, where you're working with stuff that A, is part of your body, but also is macroscopic, so you can see it. And you can imagine it, right? Even if it's the muscles under the skin that you would have to like peel the skin off to see, you can still imagine it. It's harder in this world of tiny things, what I call the tiny world. So it's helpful anytime you're starting to feel overwhelmed or a little bit abstract, or you're starting to lose your train of thought, to ground yourself in visuals of this world, because it is tangible and it is visible, but just not to us, right? We can't feel and see it, but it does exist and it is physical and three-dimensional. So zooming into that world whenever you're feeling a little bit like, oh, I've just done some metabolism, like I don't even know what molecules are anymore, is helpful. And so here's a link for you to do on your own, and it's a zoom in animation. So to get you zoomed into, like you zoom in from our visible world into this tiny world that we can't see with our eyes. Just to give you a sense of the math of the scale, one of the largest microbes is an amoeba. So these eukaryotic protozoans. This one is typically around 400 micrometers, just under half a millimeter. So we actually would see it as a tiny speck. We still do consider this microscopic. Yeasts and red blood cells are among the smallest eukaryotes. So this is the human cell that's the smallest, about 10 micrometers in size. Yeast can be actually slightly smaller than that. They're single-celled fungi. At the bacterial level, down to the prokaryotes, we have like bacteria like E. coli, usually around 1 micrometer in size on average. And then we have 10 times or more smaller than that, we have our viruses like the flu. So before I answer this question, I want you to answer it. How long have we known about microbes? Now, if you didn't do the prep, you won't know what my answer is, but that's just fine. Put your best guess. If you were just like, gun to the head, not trying to, trigger anyone about our current gun situation in the United States, but gun to your head, what would you say? Just put that into the chat. Great. Fabulous. Yeah. So before we talk about how long we've known they exist, let's talk about how long they've actually existed, which is billions of years. Okay, so way before we were even invented, they existed. It's only been about 4 centuries since we knew about them. And it's only been about 200 years, half that time, when we thought that they mattered. Now, of course, we understand that they're vital to many things that touch our lives, from being an important part of food production and manufacturing, to being sort of like the underbelly of our ecosystem, to being important pathogens that cause a lot of damage and suffering. In many ways, they have significantly shaped human history and the physical world in which we live. And this was true long before we knew about them. And it's sobering to say this, but it's true that while they would live happily without us, if all macroscopic life, like the things you see with your eyes and think are cute, like lions, tigers, bears, seals, other humans, babies, whatever, if they all just died tomorrow, the microbes would be fine. But if it was the other way around and all microbes just died out, we would very quickly be in a bind. So life as we know it is because of and dependent on microbes. Okay, so the first little kind of review we're going to do is, here are a selection of some of the scientists that you need to know about for the test. Not all of them, but just some. So Jenner, Van Leeuwenhoek, Cook, Fleming, Pasteur, Lister. I'm going to show you a contribution that one of these scientists made to the field of microbiology, and you're going to write into the chat who you think did it. So who is credited with being the first to name cells or coin the term cells? Great. So this is sort of basic from cell biology. It was, in fact, Robert Hooke. Now, which of these individuals is credited with pioneering the use of antiseptics? He was a battlefield surgeon. He had heard about the possibility that microbes cause disease, and he started to think about using chemicals to disinfect wounds instead of just, chopping things off and calling it a day. That's right. That was Joseph Lister. Famously, we still have him in our culture in the product Listerine, which is a mouthwash. Phenol, the compound he was using, is very dangerous and can cause chemical burns, but that's better than dying of gangrene. But phenolics, which are derived from phenol and are non-harmful, are still used in oral antiseptics. Which one of these individuals discovered the first antibiotic? Indeed, that was 1928, Alexander Fleming. And what was that antibiotic? For extra credit and, well, no credit really, just bragging rights today. Indeed, it was penicillin. Penicillin produced by a fungus called penicillium that killed bacteria on a plate that it was living on. Which one of these individuals is credited with pioneering vaccinations? Although it is also important to note that a form of vaccination was happening for thousands of years. For example, in China, ancient China was doing smallpox vaccinations, but not exactly the same way as he did it. That's right, that was Edward Jenner. He did it. He did this very dubiously ethical experiment, which we wouldn't be able to replicate today, but nonetheless did show that it is possible to protect people from smallpox by administering something first to trigger the immune system to protect you. First to observe living cells. Now this is debatable, but let's, what does the history say is the first person to observe living cells? Yep, the last person on the list that we haven't, well, not second to last person we haven't talked about yet. So that was Antony van Leeuwenhoek, who was a fabric merchant, made microscopes on the side as a hobby just for fun, but he made amazing drawings, had really high resolution microscopes for his time. And which one of these scientists was the one who provided strong evidence for biogenesis being also applicable to microbes? People had sort of accepted it was true of macroscopic life, that life comes from life, but they still thought microbes were maybe a special case. Yeah, that was Louis Pasteur. Great. Okay. Now that we've sort of warmed up, this is a series of lists of scientists. So they're groups of scientists that when considered together worked on something. Now, I'm not saying that they worked at the same time in history or worked necessarily hand in hand. That's mostly not the case. Some of these experiments span several hundred years. But when we look at what they did, they all can be argued to have contributed to the same thing. So I'm going to cold call you for this one. I'm going to give you a second to look at these lists of scientists and see if you can think about what this group might be they might have in common in terms of what they did. So just have a look and think about it for a minute. Okay. So now I'm going to cold call you. And before I do that, I want to remind you and reiterate the rules of cold calling, which is that you are always welcome to and encouraged to pass on an answer when you don't feel comfortable answering for any reason. Okay, so there is going to be maybe one exception to this today, and I will let you know what that is. But for the most part, when I ask you a question in a cold calling situation, it is completely fine to say pass. And just know that I appreciate it when people do that, because it gives other people in the class permission and they can relax a little bit and not feel so stressed about being called on potentially. All right, so with that said, let me see who my first victim is going to be. Let's start with Alyssa. I'm going to show you this contribution. So which group of scientists would be best described as having all together debated the origins of life, the theory of biogenesis versus the theory of spontaneous generation? Alyssa, what would you say?
I think I'm gonna pass.
That's absolutely fine. And thank you for being the first brave soul to do that. That's so kind. And now other people can kind of take a deep breath and relax and go, oh no, it's really okay. All right, we'll go to Andrew. What would you say?
Would say Reddy, Needham, Spionzi, and Pasteur.
That is correct. Yes. In a 200-year span, these scientists did a variety of experiments trying to figure out whether life comes from life or whether it's spontaneously generated. My favorite part of the spontaneous generation theory is that mice come from laundry. That turns out not to be true. But okay, next one is proposed and tested the presence of endospores. And for this, I'm going to go to Ariane. Do you have a guess or do you want to pass?
Can I pass? Because I don't know.
Yes, you absolutely can. That is always fine. Next, I'm going to ask Bimmy, what would you say?
I think it's Cohen and Tyndall. I hope I'm saying that right.
That is correct. In the 1800s, people were trying to replicate Pasteur's work, actually. He had basically conclusively shown that microbes also obey the life gives rise to life, and they're not spontaneously generated. But some people couldn't replicate his work, and they were like, gosh, what is going on? So this prompted Tyndall to propose that there was heat-resistant microbes, and Cohn later found out that there were endospores just a few years later. Okay, next up we have a group of scientists that investigated the germ theory of disease. That's a kind of a broad thing. So let's go to Charisse. Do you have a guess or would you like to pass?
I would say that was that Jenner Ehrlich?
So that there could be a case made for that. And I would not say that that is wrong. There's a better answer, but that is not a completely incorrect answer, as we'll see. So thank you for that guess. Let us go to Clara. Oh, Clara, I think that's I'm saying your name wrong because I know that your first name is different, but the screen name says Clara Stone, and that is actually, go ahead and unmute and tell me.
Yep, that's Courtney Stone.
Courtney,
Sorry,
thank you.
no
worries.
Taken over my account somehow on my computer. I got to remember to change that.
Yeah, go ahead. Who do you think? Who do you think investigated?
I think it's is it Bossi, Semmelweis, Pasteur, Lister, Koch?
Yes, that is correct, in the 1800s. Okay, great job, everyone. Let's move on to this one. This I'm just going to have you guys do in the chat. First to observe cells, what group do you think? This should be an easy one. We just talked about it. Yeah. Yep. Indeed. Okay. In the 1600s, Robert Hooke and Antoine van Leeuwenhoek were important in sort of setting the stage for we understand cells exist. Okay. Now, who developed treatments for disease? We're getting close to the end. So for this, I'm going to do a raise your hand for anyone who wants to answer this one. Which group do you think developed treatments for disease? Go ahead, Caitlin.
It was Jenner, Ehrlich, and Fleming. Jenner was the one who did the vaccines.
Yeah, good. And so we had this guest earlier for a different prompt, but you can sort of see how they overlap, right? They investigated the germ theory of disease, developing treatments for the disease. There's some overlap there. So good job, everyone. Okay. Now we've talked about this recently and 30 seconds ago, but you might not remember it. Maybe you haven't covered it yet. But I'm going to ask you about this. I talked about it in the video lectures. So Pasteur did a series of experiments with swan neck flasks, you may recall from the video lectures, where he was basically proving that spontaneous generation is still not the way that microbes arrive in the world. And what happened was people were like, okay, that's very convincing, but let's try to repeat it. And sometimes they couldn't repeat his results. So if you recall this story, can you tell us a little bit more about what his result was and why people couldn't always repeat it? Raise your hand if you have, if you feel comfortable sort of sharing that. Let's go to Nicole. Go ahead, Nicole.
So I think I saw somewhere in the book, it said like they were using different microbes, like one of them used yeast and then another one, I forget what it was, but one of them was an endospore, so it could resist the boiling.
Yes, yes. And it looks like Hannah had her hand up as well. Is there anything you'd like to add, Hannah?
I think that he said Pastor said something along the lines of like that there's microbes that are present in the air or something.
Yeah. So you'll remember or you'll review when you go back that what Pastor did that was so elegant is people had accepted, because of the meat and fly experiment, they had accepted that, oh yeah, macroscopic life that we can see with our eyes, that comes from other macroscopic life. We accept that. But they said no, microbes were different. And so he did an experiment to prove that wasn't the case with these swan neck flasks, where he bent the neck of the flask so that air could get in and out, but microbes couldn't settle directly into it. And what he showed was that as long as he boiled it ahead of time, and then he let the air in and out, but the microbes couldn't come directly down because the swan neck flask had kind of a sideways bend to it. Then they didn't grow microbes. But if he broke the flask so it was directly open to the air and the air could settle directly from overhead, then things would grow. However, when people tried to repeat this, things would grow in the swan neck flask that was unbroken as well as in the broken one. And the reason for that, as was just mentioned, is that the microbes they were using were different. So Pasteur was using yeast, which is heat sensitive, and the other people were using hay infusion, like literally taking hay and making soup out of it. And that contains soil microbes, they didn't know that, that have endospore capacity. They can make endospores that survive high temperatures. Okay, great, Linus. I'm glad that answers your question. Okie dokie. Next. From your prep and hopefully from cell biology, you may remember the 3 domains of life. What are they? Put that into the chat. Great. Yes. Oh, and I see a hand up. I'm so sorry. Because there's no last name, Maisel is what I'm seeing on the screen.
I was sorry. Can you hear me?
I can, and sort of pronounce your name for me again.
Oh, yeah, Mason. So I was gonna say bacteria, archaea, and eukarya.
Oh, perfect. Yes, thank you, Maisel. I appreciate that. Yes, you're absolutely right. Bacteria, archaea, and eukarya are the 3 domains of life. You can see that here. So before we talk a little bit more about them, which of these groups of organisms contain microbes? Go ahead and put that into the chat. Yes, everyone is right. It is all of them. So let's talk about that for a minute. So bacteria are single-celled prokaryotic cells, so the smallest type of cell, right? And archaea are also single-celled prokaryotes. These 2 groups are so similar visually, you can't really distinguish them under a light microscope. They look the same, they're the same size, they all have kind of, the different species have different shapes, but the shape types are similar. So different species of archaea look like different species of bacteria. There's not really an easy way to distinguish them visually. The only reason we know they're different from each other is most recently when people started doing biochemistry on the cell wall. They saw different chemicals and genetic sequencing. The series of DNA letters is different. That's why they're separated from each other. Now, eukarya have, of course, macroscopic organisms like ourselves, but they also have a group of protists that include algae and protozoa that are microscopic. And the macroscopic group fungi includes some microscopic members like yeasts and molds. Are there any non-living microbes? I wouldn't be asking if there weren't. So what are they? Right, exactly. Viruses, viroids, and prions. That's correct. So here's a nice little schematic summarizing all the things and kind of reviewing all these different groups. Let's talk about these different groups of microbes. Bacteria, archaea, protozoa, algae, fungi, and not all of the fungi. Of course, some are mushrooms in the forest, but there's unicellular members like yeasts, viruses, viroids, and prions. Okay. Maisel, did you have a question or comment, or were you just answering a question?
Yeah, I don't have any questions.
Okay, great. Just checking, making sure I wasn't ignoring you. Awesome. Okay. So we're going to do something just for fun now. And just to be very clear, this is not information that you have to memorize. It's more trying to help you to understand and solidify the relevance of these different groups. So some of the microbes in the world cause disease. Here are diseases that are microbial in origin. And the different possible categories of causative agents are shown at the bottom: bacteria, viruses, protozoa, and other, because there are other things like fungi and prions and things that can cause disease. So these are just the 4 options that I'm providing here. And what I'm going to do is cold call you, and you're going to tell me what you think causes this disease. Now, here is the one time you cannot pass. And the reason for that, I will sort of explain more on later, but I will say up front, this isn't graded. I'm not expecting you to know it. Nobody else in the class should expect you to know it either. It's not part of our curriculum. You don't have to know it on the final. This is more for fun and to see what our assumptions are. And I guarantee some of them will be wrong. So just know that ahead of time. And I have other reasons too, but let's just play this like a game. No stakes. I'm not going to even remember if you got this right or wrong, because I have a 2-year-old or a 3-year-old and an infant. So I have no memory of like 10 minutes ago. So I'm going to cold call people and ask you to guess for each of these. So I'm going to start with typhoid fever. And I'm going to ask Alina to guess which of these options is the causative agent for typhoid fever.
Actually, I don't know. I guess virus.
Great. Okay, that's a great guess. Salmonella. We're next going to go to Elena. Elena, go ahead for salmonella.
Do you mean me again?
Yeah, no, Olena, not Alina, sorry, Olena.
Okay, salmonella. I even don't know.
And just take a guess, like imagine you're at the casino and you put $50 down, you just got to throw the dice. Pick one, doesn't matter which one it is.
Smallpox, maybe?
Oh, salmonella is the disease. So which of these do you think is causing it? Protozoa, virus, bacteria, or other? What do you think?
It's a bacteria.
Great. Okay, fantastic. Wonderful. Moving on to mad cow, we are going to ask Aminata. What do you think causes mad cow?
I think it is a virus.
Great. Okay, we're moving on to malaria now. Malaria, we're going to ask Anastasia. What do you think causes malaria?
Bacteria.
Great. Now we're going to go to Braden for the flu.
Virus.
Excellent. So now we're going to Erica Marsden for rabies. Are you there, Erica?
Oh yeah, sorry. I usually use the space bar to unmute myself. Is it a virus?
Great. Okay, so now we're going to polio, and we're going to ask the other Erica. Erica Lowry, if you're there, go ahead.
I'm gonna guess a virus.
Great. So now for the common cold, Haley Turner.
I'm gonna guess bacteria.
Great. Now for AIDS, we're going to go to the other Haley, Haley Kronberger.
A virus.
Great. For syphilis, Hannah, what would you say?
I would say a bacteria.
Great. For herpes, we're going to go to the other Alina. So we have 2 Alinas. One of them's already talked and Olena talked. So the other Alina who has not talked yet. What would you say herpes is caused by if you are able to unmute?
Hi, sorry. Is it bacteria?
Great. Now we're going to go to Brooke and ask her what causes gonorrhea.
I'm gonna go ahead and say bacteria.
Fabulous. All right, smallpox, John, what would you say?
I'll say no, I'll say virus.
Okay, great. Jordan, you're up for pertussis.
I'm gonna say virus.
Okay, great. And Katie, you get tetanus.
Bacteria.
All right, now, first of all, I want you to take a second. When you were listening, not on the hot seat, was there any guess that you thought was incorrect? Put in his chat, yes or no. Anyone that you thought was incorrect? Okay. People are saying yes. Was there any guess that you thought was correct? Okay. Now I want you to think about what your guesses all were, and I'm going to reveal the answers, and then I'm going to tell you why we did this since it's not part of your curriculum per se. So the first thing I want to say about this is that I'm trying to generate a sense of safety around ignorance, okay? Or a sense of letting go of I am supposed to know this. We're here to learn, and it's a barrier to learning if you're afraid to learn, right? And so I want to remind you that all of these diseases, once upon a time, nobody had any idea what caused them. Very smart people had to figure it out, and they didn't know at the beginning. So they found out by asking questions, and they were not afraid to be wrong, and they were often wrong in their guesses. So I want you to try to cultivate that attitude of not being afraid of being wrong and being open to learning and sort of setting your ego aside a little bit. And just kind of getting over the anxiety around it, which is very common, right? Okay, so that's the first reason. The second is it's just fun to understand that what we're learning is really relevant to stuff that you've actually heard of before. Here are the answers. Write into the chat if there was one of these that surprised you when you look at it, the whole totality of it. If there's one of these that surprised you, what was it? Put that into the chat, or the most surprising one if one of them did surprise you. Put it into the chat. Okay, Maisel says rabies was a surprise. Keep them coming. Yes, malaria was a surprise for Alina. Stephanie says typhoid fever was a surprise. Mad cow was surprising to Aminata. Brooke says tetanus was a surprise. Melissa says rabies was a surprise. Mad cow for several people was surprising. And then someone asked about what causes mad cow. Thank you, Maisel. That's a great question. It is prions that causes mad cow. It's a prion disease causing neurodegeneration. Pertussis was a surprise to Caitlin, and you're not the only one. And yes, excellent. Okay, good job, you guys. So I just hope that's one of your takeaways from this activity. All right, so now here are the 8 groups of microbes. I'm going to give you some time. If you've done the prep, this will be easier for you, but I'm going to give you a few minutes to jot down some notes about these 8 groups because I will be cold calling you. And I want to remind you that if you're incorrect in any of your answers, or you don't know everything, you don't remember anything, or you just want to straight up pass, that is going to be fine. However, I'm going to give you a little prep time to remind yourselves as much as you can about the size, composition, structure of each. What are the features that are important about these groups from your prep? So what is their cell wall made of if they have one? Do they have a nucleus or not have a nucleus? Are they prokaryotic or eukaryotic? Do they live on land or water? Do they do photosynthesis or not do photosynthesis? Is there anything unique or important or special about them? Okay, so that's what you want to just sort of remind yourself of, those key components as well as how big they are. And in a few minutes, I'm going to cold call you, but I'm going to give you some time to prepare for this. Okay, so off you go and prep for a few minutes. All right, everyone. Let's do this. Okay. So I'm going to cold call people in the room, and I may cold call more than one person, just kind of, I'll usually ask if there's anything anyone wants to add at the end as well before we move on to the next one. But we're just going to begin at the beginning, and we're going to start with bacteria, and I'm going to ask Yusuf to tell us what you know about bacteria.
So I said that let's see. So I said that they're unicellular. They're small in size, and they usually don't have membrane-bound organelles.
Yes, that's all correct. Good job. Let's see. Just raise your hand if there's anything anyone wants to add. Let's do it that way. So I'm just going to check in about that. If anyone wants to add anything that hasn't been said, Charisse, what would you like to add?
Their shape are commonly like rod, spherical, or spiral.
Yeah, they have these sort of, each species is going to be different, let's say, than other ones, but there's commonality. Some of them are rod shaped, some are spherical, some are round. Nicole, what would you like to add?
I was gonna say that they have peptoglycan in their cell wall.
Peptidoglycan in their cell wall, exactly right. That is an important distinctive feature of the bacteria. Good job. And Haley Kronberger, what was the thing you wanted to say as well?
I was going to add, with having no membrane-bound organelles, they also have no membrane-bound nucleus. They have
That's correct.
nucleoids.
Yes, that's right. And Maisel, was there something you wanted to say as well? Maisel, are you able to unmute?
Oh, sorry. I was just gonna add that the bacteria get their energy through photosynthesis.
Some of them do. Yes, not all of them, but some of them absolutely do. Yes, good job. Mabinto? Oh, it looks like you put your hand down because that was.
Oh, I was gonna answer.
Go ahead.
I was gonna say they multiply using binary fission.
Yes, that's correct. And Caitlin, what else would you like to add?
That some of them may be able to move with a flagella.
Correct. Okay, good job, you guys. All right, next we're going to go to archaea. And for archaea, I'm going to initially cold call Stephanie. What did you write down about archaea?
Yeah, I put they're similar to bacteria in that they're prokaryotic. Their shape, size, and appearance. They multiply by binary fission. Then they may move with the flagella and have the rigid cell walls. But they're different in that they lack the peptidoglycan.
Yes, that's
and
right.
they have some genetic differences.
Yes, good. Yeah, they're very similar to bacteria, impossible to visually distinguish, but their major distinction is their chemical difference in the cell wall and the genetic differences. Good job. Sarah, I'm going to go to you for fungi. Are you able to unmute?
Yeah, so they're single cells, and then they have cell walls. They get their energy. They have organic materials energy. Yeah.
Yes, excellent. Some of them are multi-celled and still microscopic, but the yeasts are single-celled. And yes, they do have cell walls. They do get their energy from degradation, heterotroph like we are. I did see some hands go up earlier, Hannah, and I can't remember who the other person was, Alina. Was there something you guys wanted to add about archaea before I move on?
I'm pretty sure, I don't know if someone mentioned it, I didn't hear it, but that they're extremophiles.
Oh yeah, some of them are extremophiles. Thank you for mentioning that. Good job. Okay. Anyone else have anything they wanted to add about archaea? I know I forgot to ask that, and I want to make sure that I circle back. Okay. What about fungi? Is there anything anyone wanted to add to the good answer that was given about fungi? Mbintu, go ahead.
Actually, their cell wall is made of chitin, isn't chitin?
Yes, that's very important. Thank you for mentioning that. And then we also have Maria. Would you have something that you wanted to add?
They are eukaryotic, non-vascular.
Yes, they're eukaryotes. That's also really important. Good job. Okay, moving on to the protozoa. Oh, and let me just actually answer this. So Maisel said, is peptidoglycan a type of protein? And Nicole says it's a chemical. So you're both right. Peptidoglycan is actually a complex molecule that's made of both sugars, carbohydrates, and protein. It's primarily sugar with a little bit of protein kind of mixed in there. And we'll talk more about that in chapter 4. Great question. Protozoa, for protozoa, I am going to ask initially, Fada, what did you write down about protozoa?
I said they're single cell but complex, larger than prokaryotes. Most ingest organic compounds, no cell wall. They're motile via flagella and I think it's cilia.
And yes, it can be flagella, cilia, and sometimes they're motile through their pseudopods. Depends on the amoeba have pseudopods, but yes, many of them have flagella and cilia. Excellent. Is there anything anyone wanted to add to that great answer? Okay, so the next person I'm going to tap is Nina for algae. What did you write down about algae?
They can be single-celled or multi-celled.
Yes, that's absolutely true. Is there anything anyone wanted to add? Aminata says she wants to add something. Go ahead.
They are photosynthetic.
Yes. Perfect. Anyone else? Looks like Jordan has a something she wanted to add. Go ahead, Jordan.
That they primarily live in water. Their cell wall has cellulose, and they're distinct from the prokaryotes.
Yes, so they're eukaryotic. Good job. And then Haley Kronberger, what would you want to add?
Oh, she already said it. I was going to say their cell wall has cellulose.
Oh, perfect. Yes, and that's worth repeating though. That is important. Thank you. Okay, wonderful. Let's go on to viruses. Now for these non-living ones, we're sort of getting close to the bottom. I'm going to do a raise your hand for those. Who wants to talk about viruses? Caitlin, go ahead.
Okay, so viruses consist of both DNA, RNA, and a protein coat. They are acellular. They infect a host cell, and all forms of life can be affected by viruses.
Great. And then the only thing that I'll say, which you may have said correctly, but I just didn't hear it quite, was they do have genetic material. It is RNA or DNA. It's never both. So good job. Next, Maisel, did you have something you wanted to add to that? I'm not able to hear you, Maisel. Did you have something you wanted to add to that? Your hand.
Oh, sorry, I keep forgetting to unmute. I was just gonna say that their core, the DNA or RNA, is made up of or surrounded by protein.
Yes, they have a protein coat around that. Good. Maria, was there anything you wanted to add?
For viruses, yes. They are actually smaller than bacteria.
Yes. Yes. Good. Wonderful. Okay. Who wants to do viroids? Raise your hand if you want to do viroids. Mai, go ahead.
Let's see, I put that they are simpler than viruses. They require a host for replication, single short RNA, no protein coat, and cause plant diseases.
Correct. Okay. Did anyone want to add anything to that? I think that was a great summary. Caitlin, what would you like to add?
Just to add on to the causing diseases in plants, there is ongoing speculation whether or not they cause human disease as well.
Yeah. And at the moment, we have only found them causing disease in plants. So we'll just wait and see if that remains to be the case. Excellent answers, all you guys. What happened to my participants menu? Uh-oh. So my participants menu just like exploded. Hold on, let's see if I can get it back. There it is. Okay, wonderful. Last but not least, prions. Who wants to talk about prions? That would be Stephanie. Go ahead.
Yeah, so they're infectious proteins, misfolded versions of normal cellular proteins found in the brain. They form aggregates disrupt cell function, cause neurodegenerative diseases in humans and animals, and are resistant to standard sterilization procedures.
Great, yes. And then was there anything anyone wanted to add to that? Let's just have, Brooke, was there anything you wanted to add? Or has pretty much everything been said?
Pretty much everything. I thought it was interesting that like one of the main ways that you get it or can possibly get it is by eating brain matter.
Yes,
yeah,
that's right. Mabintu, was there something you wanted to add?
Yeah, I was gonna say they consist of only protein,
Yes.
no DNA or RNA.
Yes. All right, excellent. So here is kind of a cheat sheet of some of these details that we just talked about. You could use this as a way to help you study, although it's obviously not complete, but some of the really important features of each of these groups to help you kind of distinguish between them. All right, I'm going to skip over the COVID-19 lookup. The slides in the slide presentation will be posted, and during your review you can do it, but it's really just think about any disease that matters to you and look up its category and try to ask yourself, well, what do I know now that helps me understand it better? For example, you know, why is it, what group is it part of? Which category of microbe is it part of? Is the causative agent, and what features of that particular microbe make it appropriately classified as that group? Okay. All right, so we are going to take our break, just an 8-minute break, just to make sure we stay on time. I'm going to see you at 11 o'clock, and we'll talk about some applications of this information called the What's This game, which some of you, if you've taken my course before, have played with me before. All right, so I'm going to see you at 11 a.m. Have a good break. I'm going to pause the recording here. All right, welcome back, everyone. We're going to do an activity to apply our understanding. So the specific details, the specific pathogens we're going to talk about, it's not important for you to memorize those. It's the process and the logic that you need to understand. Okay, so I'm going to give you a scenario. A soldier stationed in Africa gets a skin ulcer, and based on taking information about the diseases in the area, what the soldier's been up to, how it looks, the Army doctor suspects one of two possible diseases is the culprit, either something called cutaneous leishmaniasis or cutaneous anthrax. Give you a little bit of information about it. Okay, here's his skin ulcer, and here are some pictures of leishmaniasis. It is caused by a species in the protozoan genus Leishmania, so it's a protozoan causative agent, and it's transmitted by sandfly bites. And cutaneous anthrax is caused by the bacterium Bacillus anthracis. The most likely method of getting it in this current modern age is handling goat skins, goat hides. So the first thing I want to do is ask you just to put into the chat what your guess is. So don't worry about the why part yet, but everybody should just weigh in. Which do you think? And this is, again, a case where you're not allowed to say pass, because there's no reason for you to know the answer. I made this up. So is the causative agent leishmaniasis or is the causative agent anthrax? What do you think? Great. So now I'm going to cold call a couple people in the chat just to ask you for your reasoning. And this is, it is not necessary for you to have had a logical reason. If you said what you said because it was just a straight up guess, you can just say that. I was guessing. If you have another reason, if you have some kind of reason for your guess, then tell me what it is. And so I'm just going to cold call a couple of people from each of the options. So let's first start with, let's see, I'm going to go to the top and just kind of randomize it. The first person to answer was Caitlin, who said anthrax. Why did you say anthrax?
I said that it looks like anthrax because it is that it has that circle and it's dark in the middle. The Leishmania, however you pronounce it, that blister looks red, but the anthrax is a lot darker.
Okay, so visual inspection. Great. My said leishmaniasis. Tell us why. What was your reasoning?
I just was going off of looks as well, but I kind of felt like the red inflamed circle around the anthrax looked a little different than the leishmania.
Great. So you also did visual inspection, came to a different conclusion, right? That's
Mhm.
very common. So, all right, let's see. I'm going to ask another person. Let's say, Anastasia, you chose anthrax. What was your reasoning?
My reasoning was pretty similar to Kat's. I kind of went with the dark color. Visually, it looked a bit more similar.
Great. Brooke, you went with leishmania. What was your reasoning?
Well, I was kind of trying to reason like how he could have gotten it. And it seemed a little more likely that he might be exposed to a sand fly bite than maybe handling goat skins, just kind of
Right.
out and about. Yeah.
Yeah. Maria said anthrax. What was your reasoning, Maria?
Because it says over here, I'm not sure, but it says skin ulcer. Usually that goes with anthrax, and the picture as well.
Okay, great. And then last person I'm going to ask, Fada, you said leishmania. What was your reasoning?
Similar to what someone said, I thought of the situation of where he would be, and honestly, it was also a complete guess too.
Great, perfect. So what we've illustrated is I've only given you information about how this is transmitted and you know, how it looks. And so you're trying to figure it out based on that. Now, as it turns out, we have people who had very good reasons based on those kinds of information for guessing one or the other. It's hard to tell. So what we're going to do now is I want you to think about this differently. Okay. And I want you to think about why this is relevant to what we've just been learning. So if we want to diagnose, and let's learn a little bit more. So remind you of what the causative agents are. Leishmaniasis is caused by a protozoan parasite called Leishmania, and anthrax is caused by a bacterium called Bacillus anthracis. So think about how you would be able to distinguish between these agents. Imagine that you have a lab and you can take tissue samples and you would look things under the microscope. You can run a chemical analysis. You can look at the size. You could look at what's inside of the cell. We could ask questions about its metabolic abilities, like anything that might help you figure out what's the causative agent. So ask me in the chat a question that you think I could answer. And try to stay away from, you know, we've just tried to make a distinction based on visual appearance and based on lifestyle and, you know, context, assuming that information's already been gathered, right? Now, what other questions could we ask that would be helpful here? And when I answer them, if you think, oh, now I know the answer based on what you said, raise your hand and I'll ask you to tell us what you think. Okay, so in the chat, put any question that you think I should answer. So Caitlin says, does it have a flagella when viewed under a microscope? Does the causative agent have a flagellum when viewed under a microscope? Let me just run that test really quick. So hold on while I find out. Okay. Okay, so this particular causative agent that caused this disease does have a flagella. And then what other questions I'm seeing? What is the shape? Okay, that's a great question. The shape is kind of a rod shaped. Nicole says, does it have a cell wall? It does. What are its size measurements? The size measurements around 2 micrometers by about half a micrometer. And then does anyone have a guess based on those answers so far? Raise your hand if you have a guess based on those answers so far that you can defend with logic based on what you've been hearing. Just going to take a sec. Okay, Erica Marston says, yes, she does have a guess. Go ahead.
Because of its shape, I want to say it's a bacteria because it's rod shaped and they have very specific shapes.
Okay, great. So you think it's a bacteria based on its shape. Nicole also raised her hand. What would you like to say, Nicole? What do you think and why?
I think it has to be a bacterium because protozoas have no cell wall.
Okay, great. And Jordan has a guess. And what's your guess, Jordan, and why?
It's kind of the same as everyone else's. It's a bacteria because there's no cell wall in the shape.
Great. And then I also see in the chat, people are saying they think it's a bacterium because of similar reasons. So yes, you guys are all correct. Now, hopefully this helped to shift the way we think about these questions, right? Because when I ask you a question on a test, if you have any moment of anxiety, you might sort of like by default shift to clinical things that you feel comfortable with, like, looking at symptomology, looking at, exposures, things like that. But in this class, I'm not asking you to memorize details about every specific organism, right? So it wouldn't make sense for me to expect you to know that. Like it doesn't make sense for me to expect on a test that you're going to know what a skin ulcer of Bacillus anthracis looks like. That's not part of our curriculum. Instead, you need to be comfortable with the logic and the information. Like, what information do you actually know that I provided that would help you to make this an answer? Okay, and then I'm just gonna say, okay, and I got a question, a message, and Arian, I'll get to that at the end of class if that's okay, and that's a great question. Okay, perfect, and just remind me if you wouldn't mind, because I may forget. So good. All right, so now we are going to move on to one other example. So Maria, a resident of Washington Oaks, which is a local elder care center, develops pneumonia. We have to diagnose her appropriately if we want to give her the right treatment. So the first question is, what types of microbes can cause pneumonia? Again, you don't have to know this, but many of you will know the answer to this. So let's start there so we know what the options are. What kinds of things can cause pneumonia? Because bacteria can, viruses can, fungi can. Those are all possible causative agents of pneumonia. So we see that they're very different, which means that if we want to treat her, giving her an antibiotic, which is only effective against bacteria, is not going to be very helpful if what she actually has is a virus, for example. So we want to know what she's got. I would like you to ask a question that would help you determine whether she has got a viral, fungal, or bacterial infection. So ask a question and then chat that you think that I could, if I answered it, it would help you to narrow this down. Okay, is there a cell wall, says Nicole? The answer is yes. Erica says, what is the cell wall made of? That is an excellent question. It is made of chitin. Does it have membrane-bound organelles? Yes, it does. Are both DNA and RNA present? Yes, they are. Okay, based on that information, does anyone have a guess that they'd like to defend? If you do, go ahead and raise your hand. And it looks like, oh, Haley, go ahead. What is your guess and how would you defend your answer?
Well, I would say first, it is not a virus. We can conclude that since there's both DNA and RNA present, that it could not be a virus. And I would say that it is fungi based off of what the information that was given, because you said there was a cell wall present, correct?
Yes, I did.
Yes, yeah. Say fungi.
Yes. Okay. So your logic excludes viruses because of the genetic material being both RNA and DNA. And you think it's fungus because of the presence of a cell wall. Does anyone want to add any information that help them distinguish between bacteria and fungi, because those both do have cell walls? Charisse, what would you say?
You also mentioned that the cell wall is made of chitin, and that's specifically fungus or fungi.
Exactly. Good. All right. Good job, you guys. Courtney, did you have anything else you wanted to add?
No, I was going to say the exact same thing.
I figured I could feel it. All right. So good. Excellent diagnosis, guys. Okay. So now I want to just write quickly into the chat. Why do microbes matter? Just anything that comes to mind. We talked about this on Monday. Maybe after this prep, you have additional answers, but just put them into the chat and I will read them out. Why do microbes matter? What is their importance? Okay, so we've got some of them ferment food for us. That is true. Some of them are important for our digestion. They are important as in our ecosystem. That's also true. Some of them live on us. Very true. And we need to know about them in order to treat disease, diagnose disease, understand our environment, prevent food from spoiling, do research. They are a part of life, like they help life to be here. They're important in industry, and they're part of our immune system. That's right. They help us to develop new technology, and that's a very broad statement that's very true. And they can help make things like industrial chemicals, for example. They provide vitamins in our body, and they're useful in vaccine production and disease prevention. Excellent work, everyone. So here are my answers. Oh, not yet. Never mind. Here is something else I want to say. So some microbes cause disease, and those that do have a massive impact on human health and death and suffering. More people have died of microbial disease than have ever been killed in war so far. But most pathogens I'm sorry, let me say that again. Most microbes are not human pathogens. So there's some of them are pathogens of other animals, but some of them are not pathogens of anything. So it is important to recognize that, because the ones that cause disease can have a sort of outsized presence in our minds, but most microbes are actually not harmful to people. So some of them are a part of our normal microbiome, which is an important part of our immune system, helps protect us actually. Some of them have important roles in the ecosystem in terms of either decomposing organic material, making it degradation products available for us, or fixation, going the other way. They create organic materials that we can't otherwise have available to us. Some are domesticated, right? Some have important roles in food production or in industry, and some have been genetically engineered to produce useful things like insulin or for research purposes. Many of them are very good. If we just study them, we can learn about other things, but they're easier to study. So what do you think? Do we know everything about microbes yet? That's right, a resounding no. We have cultivated less than 1% of all species because we don't know how to feed 99% of them, right? Can't grow them in a lab. Why do we know they exist? Well, partly that's because of genetic sequencing. So it's this kind of a new idea that we know so little. Just as one little tiny example, here is a region of the Sargasso Sea known as the Bermuda Triangle, and a little sample of this ocean. There were 2,000 new bacterial species found, revealed by genetic sequencing. So any one of them has potential utility in all kinds of ways for us. And as we feel, I think, most keenly today, there are many challenges that are related to microbiology that remain to be addressed. Okay, so the field of microbiology has been helpful in several sort of like dramatic ways. For example, smallpox has been in the human population for as far as we've got written records, and presumably before that, and caused millions of people to die. In 1977, understanding smallpox led to development of a vaccine that eradicated it from the earth, which is amazing. The Black Death has periodically reared its head throughout history, and at one point in the Middle Ages, it killed 1/3 of the population of Europe. Now well controlled with antibiotics, not gone, but controlled. If you get the Black Death, which is the bubonic plague, or Yersinia pestis infection, you can get an antibiotic and be cured. Despite these advances and many others, it's still true, soberingly and sadly, that around the world, most of the diseases are impacting children, and a lot of children die every day from just respiratory infections and diarrhea. And there's plenty of things to worry about in this country, not just in parts of the world that you might consider to be, to have fewer resources. And in particular, of course, we've all just been through a couple of years of the pandemic, which is something that we've always worried about and has happened before and may happen again, just a different disease. There's new diseases emerging all the time, sometimes because a bacteria we previously had well controlled is reemerging. It's developed resistance. Tuberculosis is one particularly worrisome example. And also because new things arise by crossing the species barrier. Okay, so what we're going to do now is another Kahoot. And so I'm going to open the Kahoot. That gives you time to do what you need to do, which is either go to kahoot.it, making sure that when you open that web browser, you can still see my screen, or you can download it onto your mobile device. Or if you've already downloaded it, I would say if you are still having to do that, you might not have time. But we're about to do a Kahoot. I'm just going to open this puppy up and get started here. And I'm going to share my screen and just a second. Okay. So here it is. And the code to put into the Kahoot is 6959979. 6959979. I'm putting that into the chat as well. Use that pin in order to join the Kahoot, but you will need to see my screen in order to understand what buttons to press on the Kahoot. All right, we've got 38 people on the call today. We've got a decent number of people logged in. Let's see if we can get more. Give you about 30 more seconds to log in. If you're not logged in yet, you are always welcome to participate in chat instead. If the Kahoot is too difficult to get into, it may kick you out, in which case the game pin is always shown on the bottom right of the screen for you to rejoin if you need to. Great. Okay, just another few seconds to let the last few people log in if you can. If you have to use the chat, that is also fine. All right, here we go. What best describes the contribution of this group of scientists, Tyndall and Cohn? Yep, hypothesized and or discovered endospores. Good. What best describes the contribution of this group of scientists, Jenner, Ehrlich, and Fleming? They developed antimicrobial interventions. Very true. Which group investigated germ theory? That is the theory that specific microbes cause disease. It was Bassi, Semmelweis, Pasteur, and Koch. Which domain contains microbial members? That's right, all of them do. Okay. A microbe 100 nanometers wide made of RNA and protein is probably it is probably a virus, right? It's too small to be a cell, doesn't have DNA or membrane for that matter. A microbe with a cell wall made of chitin is probably A fungus, right. 0.45 millimeters is equivalent to what? Correct, 450 micrometers. So millimeters are 1,000 times larger. So we actually multiply this by 1,000 to get the micrometers. True or false? Most microbes are pathogens and cause disease. Is this true or is this false? It is false. Most microbes are non-disease causing in humans, at least, although there are certainly some that do cause disease. Let's see who made it to the podium today. If you were not on the podium, just know that I know that you were right behind these fast front runners, and congratulations to everybody for their excellent work today. Okay, let's go back to our slides. So I have a few things I want to talk to with regard to lab. The unknown ID project first, a quick review of the lab 1 calculation that you'll need in order to finish the lab 1 stuff that's due Sunday. And then a preview of lab 2. For some of you, the lab 2 pre-lab is due before we meet. That's if you're in my Saturday class, or it's due like Monday night. So you kind of want a little heads up. Before I move on, I just want to check in. Courtney, your hand is still up. I think that's from before. I just want to make sure I'm not ignoring a question that you have.
I forgot to take it down. Thank
Perfect.
you.
Just making sure. Okay, let's talk about the N90 project for just a second. So it's a major project. It's going to take you probably somewhere between 6 to 10 weeks to finish. And the first thing you have to do is do this Sunday, and it's very easy. You just go to the unknown ID sign up page and sign up for a number. That's all you have to do by Sunday. But if you've got some time, read about the project so you can kind of get a leg up. Really quickly, what are you going to do next? Once you have this unknown ID number, it hooks you up to a particular bacterial species, but you don't know what it is. I don't know what it is. The only person who knows is the technician, to Win. And she is going to be your primary contact for this project. So once you've got this number, go ahead and read the instructions for part 1. It's going to basically entail emailing the technician too, and requesting a picture with the gram status, or not, a picture of a gram stain of your organism so that you can determine its gram status and morphology. You might want to hold off until Monday to do this, just because I'll be still getting organized over the weekend, and she may not be ready for you yet. But starting Monday, you can start this process, and it shouldn't take very long. Like, you're going to send an email, you're going to get a picture back, you're going to interpret it, you're going to use that to pick a dichotomous key, and then you're going to submit your very first part of this project to a Dropbox. After that, you're going to use a dichotomous key to do between 2 and 4 tests that will lead you to your group ID. So again, you're going to send her a request for a particular test. She's going to send you a picture, you'll interpret it, and that will lead you to your group. And the group is going to be something like a dozen or 2 dozen or 100 or 200 organisms. Different bacterial species, you have to narrow down which one you have. That's part 3. This is the part that's going to take most of your time. And so again, you're going to decide what tests to run. You're going to send away for a test. You're going to get a picture back and use that to narrow down what you've got by eliminating the species that are definitely not yours until you have just one left. At that point, you're going to write a lab report about it and submit it at the end of the quarter. And that's the project in a nutshell. So this week, unknown ID sign up. The first part, determining your gram status and morphology, is not due till week 3, but highly encourage you to finish it by the end of week 2 if you can. So by next, not this Sunday, but next Sunday, that's your goal. So Monday would be the earliest time to send out your email to the technician saying, I am so-and-so, here is my unknown ID number, please send me a gram stain. Week 5 is the deadline for part 2, knowing what your group ID is, but it is possible and encouraged to get this done sooner, which leaves you more time to figure out your species ID. Okay. Okay, now I do want to revisit for lab 1, you have to do some calculations determining how much microbial load there is in water and air samples. I want to do a quick refresher with you. It also illustrates how you can practice this on your own. Okay, so this slide I had in my last slide presentation. Let me just go back and do that. But we're going to modify it. Okay, so I would like you in the chat to give me a number that's close to 100 or close to 85, somewhere in there, but is not exactly 100 or 85. So if you were to count this, what number do you think you'd come up with? Okay, we've got 90. 90 is a good one. Let's do that. The area in the example I've generated is 1/4 of a square foot. So give me a different number, 10th or 15th or a 20th or a 5th or a half or whatever. Give me a fraction of a square foot, half. Okay, excellent, half of a square foot. If this plate was 1/2 of a square foot, which would be huge, then how many plates would we need to sample to get a whole square foot? We would need 2. Okay, since we don't want to do that because we want to save resources, since our area is half, we want to actually multiply the number that we get on this plate by what in order to account for the fact that we did not sample a whole square foot, but only 1/2 a square foot. Yeah, we want to multiply it by 2. That's right. So we want to multiply it by 2. Okay, our exposure time. Give me exposure time in minutes. 20 minutes. Okay, 20 minutes. Exposure time of 20 minutes. 20 minutes is what fraction of an hour? 20 divided by 60 is 1/3. That's right. Okay, so what do I have to do to this number, 90, in order to account for the fact that I want to know what would have fallen in a whole hour, but I only sampled for 1/3 of an hour? I have to multiply by 3. Yeah. Okay, so I've just adjusted all these numbers and you see everything's worked out. Both of these ways of doing it are perfectly legitimate. If it's easier for to remember all the units properly, you can take the CFUs, 90, divided by the product of area times time. The time was 1/3 of an hour, the area was 1/4 of a square foot. This is going to be 1/12 square foot hours. And then you would take 90 divided by 1/12, which is the same as saying 90 times 12. Or you can do it this intuitive way. If you just understood the logic we did earlier, take 90 times 2, because we know we only sampled 1/2 a square foot, but we really want to sample a whole square foot, and multiply by 3 because we only sampled 1/3 of an hour, but we want to know a whole hour. And this answer we get, what is the answer? This is not the correct answer. What we get is, great. Quick check-in. Does this make sense to you? Yes, no, or kind of. Put that answer into the chat. Let's have everybody weigh in. Don't be ashamed to say if you still don't get it, that's completely fine. And I invite you to stay behind at the end of class if you want to talk about it some more and do another example. I'm happy to do that. But I'm providing this as an illustration of how you can practice this yourself just by manipulating these numbers. All right, we're going to move on to water samples. Okie dokie. So again, here is a plate. Give me a number that's not 20, but is sort of similar to 20. 25. I love it. Okay. Instead of 1/4 of a mil, let's do something like 1/2 a mil. Usually we do 0.1. That's much more common, but we're going to manipulate this a little bit. And instead of 1 in 100, give me another dilution. Okay, 1 in 200. Okay. So 2 ways to do this math. What we want to know is how much stuff is in a milliliter. We didn't put a whole mil on our plate. So we know if we put a 0.5 mil, what fraction of a milliliter is that, guys? It's 1/2, right? So we already know that we're going to have to do what to this number? Since we only sampled 1/2 of a milliliter, we're going to have to multiply it by 2, correct, right? And we diluted it 200-fold, okay? And so what do we know we're going to have to do to account for that? We're going to have to multiply it by 200. So we could take 25 divided by the product of the volume times the dilution. That's going to give you, sorry, this is 0.5. That's going to give you 1/400, so 25 by 400. And let me just adjust this and say 0.5 mil times 1/200 is going to be 1/400, and 1/400 I believe is actually 0.0025 if I'm not mistaken, but someone can run that calculation really quick. 1/400 is 0.0025. Yes. Okay. Or we can do it this intuitive way. We can say we know that we have to multiply by 2 because we only sampled 1/2 mil, and by 200 because we diluted it 200-fold. Either way, we're going to get the same answer, which is what, guys? Yep. Okay, let me check in again. Yes, no, or kind of understood this. Great. So feel free to stick around after class and ask me to go through it again, or any particular part of this, or email me, or play with this slide until you feel more comfortable and confident. You do have to know enough about it to answer the stuff that's due Sunday, but we're going to do this again for Lab 2 as well. Okay, there is a CFU Kahoot. I don't think we have time for it. I'm going to save that for the end, though. If we do have time, I'm going to come circle back around. Otherwise, I may assign it outside of class as a practice. The reason I want to move on is I want to talk to you about resolution. So next week, our topic is microscopy, and the Lab 2 will focus on that as well. So we have to understand what it is and how to calculate it. Resolution is basically how much detail you can see. A really good definition of resolution is in microscopy that's helpful is the smallest thing that can be seen with that equipment. Okay, so for example, the resolution of the human eye is 200 micrometers. That means that if something was 200 micrometers or larger, we'd be able to see it as a speck, but if it's smaller, it's going to be invisible to us. The light microscope is 1,000 times better, right? It can see things that are as small as 200 nanometers or 0.2 micrometers. And the electron microscope gives us a ton of detail because it can see even better than that. It can see things that are even smaller than 0.2 nanometers. So it can see viruses, and it can see even like large molecules like DNA and membranes and stuff like that. The light microscope is really helpful with cellular things because all cells are going to be bigger than this. And our eyes useful for seeing insects. That's about it. So what these numbers are is something called resolving power. Resolving power, you can calculate it. And what it means is literally means the smallest thing that can be seen, right? So given that resolving power can be calculated and that these are the numbers, okay, is it better to have a piece of equipment with a small number or a large number for its resolving power? What do you want, small RP or a large RP? Put that into the chat. Yeah. So in this case, smaller is better. And to illustrate that, if you're feeling a little unsure, and if maybe it's just a semantic thing, these are the resolving powers, okay? 200 picometers, 200 nanometers, 200 micrometers. Which of these resolving powers would you like to have? Yeah, you would want this one. And this is the smallest thing, right? That the electron microscope can see the smaller things that are this small can be seen with an electron microscope that would not be visible with a light microscope or a human eye. So the smaller the RP number, the better. Okay. So here is the, whoopsies, my animation's getting out of hand here. Sorry about that, guys. Here is the equation for calculating resolving power. All you need to know is two things: the wavelength of incident light or the illuminating thing, usually visible light, but it could be ultraviolet, which is not visible. It could even be electrons, which have a wavelength and they're not visible. So the wavelength of whatever you're shining on the sample divided by 2 times the NA, of the lens. This is just an empirically derived constant that is a it's a product of that particular lens. We can calculate it, we figure it out. There's no units involved. It's just a numerical aperture. It tells us something about the resolving power of that lens. So let's start with what wavelength is. Visible light, actually on a spectrum, the EM spectrum, the wavelength of the blue shifted light is shorter compared to the red shifted light. And then below that, smaller yet, is ultraviolet light. And then way over here is electrons. So which color of visible light is going to give you the best resolution? Yeah. It's going to be on the blue shifted side because the wavelength is smaller. Why? The smaller the wavelength, the smaller the numerator part of this fraction, which means that the RP is going to be smaller when you do the calculation. To get our smaller RP, you want a shorter wavelength. Okay, so if we're talking about visible light, you want it on the blue side. If we're talking about UV light, that's going to be better. And if we're talking about what's the best, electrons are going to be the best. Now, as far as numerical aperture, it is a unitless number that's just going to be given to you. You don't have to memorize it. If you look at an objective lens on a microscope, you'll see it reported there. Right there, you can see it. Okay. And so in this equation, do you want a lens with a large NA or a small NA? If you want this to be small, right, you want your RP number to be small so you can see small things. Do you want this to be bigger or small? Yeah, the bigger the better, right? So you want your NA to be big. And you can see a couple different lenses with some typical NA values. The specific NA value depends on exactly who made the lens. But what you can see is that the NAs sort of max out around here. Okay, so here's just an example. And it's similar to something you'll have to do for pre-lab 2 and that we'll talk about in lab next week. So imagine that you have a microscope and it's got a 15x ocular, it's got an 80x oil, it's got 100x air. So this is going to be the eyepiece you look through. You have then the choice of either sliding this into place or this into place. And you also have a filter that allows you to use yellow or green light. Okay, so let's dispense with this first question. Let's not worry about it. But which of these two objectives would you think would give you better resolution? Which one do you think would give you better resolution? Great. Awesome. So we're getting, I would say, like a 60-40 split between people who think the 100x is better and people who think the 80x is better. Now, I will tell you the answer, but first I just want to say thank you for answering, and don't worry about it if your is not the right answer, because you've got really good company. Lots of people had the same answer as you. So it turns out the better one is the 80x one. If you feel confident defending why you chose 80x, raise your hand. Why did you make that choice? Andrew, go ahead.
I originally chose a 100x because I was going based off of the 100 versus 80 number. So 100 would be bigger, but that's not what actually goes in the resolving power equation. It's actually the NA number, and the NA is 1.2 with 80 versus 5.
Isn't that interesting, guys? So yes, it's easy to pick this 100x because the magnification is clearly going to be better. But we can see that the resolving power is not better because we need an NA that's large, and the largest NA is provided by this 80x oil. So that's an important thing to pay attention to. Okay. Now let's choose a light source. Which of these two light sources? Put into the chat which one you want to use, yellow or green. Right, everybody's choosing green. Raise your hand if you want to tell me why'd you choose green. Go ahead.
I was just saying because it's closer to blue.
Yeah. Anyone else want to give their answer? You all are right. You're all agreeing. If you had a different logic or you want to add to her logic in any way, Haley, what would you like to add?
It has a shorter wavelength, so it'd be easier to see because you want it to be smaller.
Yeah, exactly. Both of you are completely right. So the closer to blue, the better in visible light. And the reason for that is that the wavelength is smaller the closer you get to blue. So you're looking at the wavelength 500 is smaller than 600. So that's going to be better because when you do the RP calculation, you're going to get a smaller number. Now remember, RP is equal to wavelength divided by 2 times NA. So when we use our ADX oil and our green light, what is our RP? Do that calculation, put that in the chat. RP is wavelength divided by 2 times NA. What is the answer? Put that into the chat. Excellent. I'm going to wait for anyone else to, good. Okay, great. Yes, so wavelength is 500 nanometers. So 500 nanometers, and make sure to use your units, divided by 2 times NA. So 2 times 1.2 is 2.4. 500 divided by 2.4 is 208. And you always want to use units, which are nanometers. And so what does that RP actually tell you? Raise your hand. Once you have calculated this number, what does it allow us to know? Raise your hand if you can tell me, what does this 208 nanometers mean? Mariceli, go ahead. You're still muted. Go ahead. Oh, yeah, there you are.
Yeah, it's because it's bigger. We're trying to see whether it's bigger or smaller.
Yeah, so it has to do with size. So 208 nanometers is a size. It's telling us size of what? Anyone feel free to hop in as well if you have sort of want to help her out here with what she's on the right track of explaining. What is 208 nanometers telling us? It is a size. What's it a size of? Erica, go ahead.
Is it like how close you're seeing it, or like the resolution power?
Yeah, it's and also Nicole's hand went up. What would you like to add, Nicole?
I was just taking a guess. Is it the smallest thing that you can see?
Yeah, that's right. And some people have said that in the chat. Katie says the smallest size the scope can see. Yes, that number is if something was that big, and this is what Mariceli was getting at. If something is that big or larger, this microscope would see it. But if it was smaller, it wouldn't see it. Does that make sense, guys? Yes, no, kind of? Put that into the chat just so I can get a pulse on it. Great. Okie dokie. So I put the answers in here so you see the math has worked and you can use that in helping you to figure out what to do with the lab. The last thing I want to talk about is what's coming up. And I have a special slide for those Saturday people. So just know if you're in Saturday, some of this is a little bit different. So hang on for a second. But for everybody, this is true. Wednesday night, which is today, is your orientation module is due. So the quiz, the lab safety quiz, lab preferences. Yesterday, I fixed a few errors in these quizzes thanks to excellent feedback from students who were finding some errors in my quizzes. Sorry about that. So make sure you have those done by the end of the day. And if you didn't get full credit yet, take them again. By Sunday night, there's the basically chapter 1 quiz. All the lab 1 stuff is due. There's a wiki for you to contribute to on Canvas, a Dropbox for you to put something into, and a post-lab quiz. You need to sign up for your unknown ID number. I would encourage you to read about the unknown ID project, though you don't want to get it started till Monday. And then Sunday night is not exactly the pre-lab deadline for everyone. I would say by Sunday, you want to have read lab 2 background and instructions pages, except for Saturday people. So hang tight, Saturday people, because I have something extra to tell you. So for example, pre-lab 2, for those of you who are here normally in the morning and your labs are in the morning on Monday, Wednesday, pre-lab 2 is due for you guys on Monday night. And for those of you who are in the night class, if you're visiting this Zoom session from the night class, Your pre-lab is actually due Tuesday night. Anastasia, that's a great question. Let me get to that in a second. And don't let me forget. Now for Saturday people, this deadline is actually sooner, and I'm going to talk about that in 1 second. Before Monday's Zoom, your job is to go to the microscopy topic page, watch the video lectures, review the slides, read the textbook, do the pre-quiz, because there's a pre-quiz, not for credit, but helpful. Do all that prep before Monday so you feel like Monday is a review session for you. You can investigate those areas that are still weak. You can solidify the things you did understand. It's like, much more fun to participate if you've done the prep. Okay, before I answer these excellent questions in the chat, there's one other thing I want to say. If you are in a Saturday lab and you're here today visiting, thank you for coming. Heads up, your pre-lab 2 is due on Friday. We are meeting this Saturday at 9 o'clock in the morning for our lab 2. So your lab 1 is still due Sunday like everyone else, but your pre-lab 2 is due Friday. So make sure you get that done by Friday and come to class on campus, Shuckson Hall 139. It is the building that is just south of the, it is just north of the southeast corner of campus. And if you look on campus, there's a map and some instructions. Okay, so with that, I'm going to answer some of these great questions that have shown up in the chat. So let's start with Anastasia, where we'll be able to find the recorded lectures. Yes. Okay, so there's 2 places. I'm just going to stop the recording here, and then I'm going to go answer all these questions. Let me just stop the recording. All right.